Okay, thanks, Jeff. So today I'll be talking about MRI biomarkers in the CHIMERA model of mild traumatic brain injury. So just a bit about TBI. So it's a very prevalent disorder. Many people around the world are afflicted with it. And for most people, actually not a big deal. Symptom resolution happens within days to weeks. But there's an unfortunate proportion of those people, 25% approximately, that actually have symptoms that persist on the order of months to years. So those symptoms can range from cognitive deficits, anxiety and depression, more likely to develop things like Alzheimer's and Parkinson's. So, you know, we're all animal MRI people, so we got to ask the question, how can we help? So animal models are a good platform for the development of biomarkers and therapies. In TBI, there's a really large heterogeneity with how the injury can happen. So we can sort of try to control for those heterogeneities. So, you know, we have continued to use animal models, but the relevance of those models are put into question. I mean, greater than 100 potential therapeutic agents that have shown promise in animal models have actually failed to enter the clinic. So this talk is really not really showing a lot of results, but really just to illustrate our thinking in terms of where we think this is going to go. So we're, in a sense, we're just drawing the roadmap now as to how MRI can fit in with preclinical TBI research. And really, you know, instead of jumping in and addressing every issue, you know, the question is, what are the opportunities that are specific to UBC, guided by locally available expertise and systems? And really, the sort of thing that ties everything together is a novel injury model that has been developed at UBC. So that model is called CHIMERA, or Closed Head Impact Model of Engineered Rotation and Acceleration. That's led primarily by Doctors Cheryl Wellington and Peter Krypton, who co-invented the system and has characterized the injury that's inflicted. So this is sort of an overview of more traditional models where you have a direct impact either on the open head or even the open brain, where you have a direct impact either with a pneumatic actuator or a fluid percussion device, or you have a weight drop that's striking a sort of disc or helmet on the animal. So what sets the CHIMERA injury model apart is the ability to model rotational acceleration. So here you have the rig here, it's driven by pneumatics, and the animal is placed supine on this incline. A piston strikes the animal from beneath the supine skull, and you basically have rotational acceleration. So this is just a video of what happens during the moment of impact. So you can see that you have the full range of rotational acceleration, both forward and backwards in, you know, something like a automobile accident, for example. So you have, we also have a version of that injury model where we put a interface helmet, 3D printed, onto the animal, and that allows a higher energy to be delivered without skull fracture. This has gained quite a bit of success. It's licensed in, I believe, over 30 institutions. It offers the ability to tune the energy, has high reproducibility, and it's scalable to a variety of different animal species, including mice, rats, and ferrets. So I just want to sort of give an overview of what's happening during the injury. During the primary injury, there is literally tearing or shearing of the tissue. So in the axons, you have disruption of the axon microtubules, and within the plasma membrane, the myelin sheath, because of the stretching, you have these pores that start to form. And in addition to that, we have the disruption of the blood-brain barrier. So what does that mean in terms of the secondary injury that immediately follows? So in terms of the axons, you basically get a cytoskeletal disruption as well as a series of axonal varicosities along the axon length, which is known as beading. And in terms of what the consequence for the poration is, basically there's a massive efflux and influx of different ions, and that sort of produces a vicious cycle of basically an inability to come back from constant neuronal depolarization. So there, the membrane pumps sort of go into overdrive, trying to restore the ionic balance, and that leads to a glucose energy crisis that leads to a greatly reduced metabolism of glucose. In terms of the blood-brain barrier, basically, if that's ruptured because of the initial mechanical insult, you have an influx of blood products, including protein tangles and other sort of species within the blood. And if you don't have a well-functioning blood-brain barrier, then the ability to clear waste products is compromised. So that leads to possibly a variety of different chronic injury conditions, such as permanent axon and myelin damage, inflammation, associated with increased astrogliosis, as well as glial activation. You have a decoupling of the vascular response in response to metabolism, and you have protein accumulation, which may trigger later neurodegenerative diseases such as Parkinson's and Alzheimer's. Okay, so what does CHIMERA produce in terms of the damage? So the one thing that it's sort of sort of characterized is the diffuse axonal injury. There's a associated with the long white matter fiber tracts, especially the optic tract, but also in the corpus callosum and the anterior commissure. Most of those areas also are associated with inflammation, and we also see inflammation in some of the gray matter areas, including the hippocampus and the brachium of the superior colliculus. In the more severe injury with the helmet interface, we also saw blood-brain barrier extravasation of immunoglobulin. In terms of behavior, depending on the energy applied, the animals regain consciousness between 6 to 30 minutes. In terms of behavioral tests, it's most consistently associated with cognitive deficits in regards to learning and memory, as well as fine motor deficits. And so far, there's not really that much evidence that it affects things like anxiety, depression, or suppression of social behaviors. So how does MRI fit into preclinical models of TBI? So what we did was we did a literature survey of the past decade. And what we found in terms of the use of quantitative MRI, over almost half of the papers were mentioning diffusion. So there was some spectroscopy and CESS studies, as well as perfusion and fMRI. But the lion's share went to diffusion, and just for DTI or diffusion weighted images alone, that makes up over half the references. So that's basically what we've started to look at first.
So this diffusion imaging is associated with looking at the white matter microstructure in these TBI models. And the other thing that we would also like to start looking at is the perfusion aspect of it, specifically looking at our arterial spin labeling and dynamic contrast methods. So not only does that sort of look at the, elucidate the vascular function and the blood-brain barrier integrity, it does serve sort of a UBC specific purpose because UBC, actually Cheryl's group developed a first-in-mouse assay of plasma GFAP, which is a marker of astrocytes. And so that's potentially a good way to validate a blood biomarker that is already used in the clinic. So let's look at diffusion. How is diffusion used to look at mild TBI? So this is an image from a paper in 2017. This was done with CHIMERA, done at the NIH. So what they found was using DTI ex vivo, they saw that the FA reduced in the areas that were pretty much associated with the histological markers of axonal damage. So they found reductions in the corpus callosum, the optic tract, and the superior colliculus. And that makes a lot of sense because in diffuse axonal injury, we have beading, which it sort of increases the freedom of movement in the radial direction, as well as disrupting the longitudinal sort of transport of water along the axon. However, in some other references, they actually observed an increase in the fractional anisotropy. And the thought there is that what's happening at the same time is that the extra axonal space is changing along with the diffusion within the axon. So as inflammation occurs, the astrocytes actually get thicker, and there's the arrival of microglia in the areas of the damage. So essentially what you have are two competing effects. You have sort of the reduction in the FA due to what's happening in the axon, but we must also consider the fact that, MRI looks at the signal from an entire voxel. So the inflammation may also play a role in kind of counteracting those effects. So what to do in order to sort of detangle these competing effects? So people have actually tried to look at diffusion biophysical models. So one thing that has seemed to gain popularity in the context of mild TBI is looking at NODI. So instead of modeling the water diffusion as a single ellipsoid, they assume that there's actually a separate compartments that separate the extra axonal space and an axonal space that is constituted of white matter fibers with some orientation dispersion. So the two main metrics are ODI, which is an index between 0 and 1 that illustrate the amount of angular dispersion. And there we have NDI, which is essentially an axonal volume fraction. So happy to say that my fellow Canadians are sort of leading the charge in this idea. So we have two papers, one from University of Toronto and one from University of Western Ontario, that have looked at these changes in a controlled cortical impact model. And they found a reduction in the ODI, or actually an increase in the ODI in the optic tract. And separately and independently, they found in the corpus callosum that there are elevations of both the NDI and the ODI. So I must note that this is in the context of looking at also changes in the DTI, and no change in the FA was statistically significant. So that sort of illustrates the value of using a more complicated biophysical model. So
this is, so we've started to look at NODI on the CHIMERA model here at UBC on our new 9.4 Tesla system. So sort of when we started anecdotally, we saw that in one injured animal, we saw a reduction in that NDI in the corpus callosum. So we went ahead and did a small study with 5 shams and 5 injures using ex vivo 3D EPI with a variety of different directions and V-values. And I really have to give a shout out to Anne Wheeler and Lisa Gazinski from the University of Toronto, who helped us set up the multi-subject registration and segmentation necessary to compare the 2 different groups. We use structure-wise comparisons, and really the only metric that survived multiple comparisons correction was NDI. So we found that in the TBI animals, they have a lower NDI than the shams. But we did note that the areas where we find these differences, they don't really fit super well with the previously regions that were found histologically and in other DTI studies. So, okay, so I just really wanted to show this one opportunity here at UBC. You know, we'd love to have a histological reference to validate our diffusion imaging and all of our different modeling that we get from quantitative MRI. So here at UBC, we actually have, and I'm sorry if this is choppy, we have available this technology called tissue clearing and light sheet microscopy. And so this allows us to look at a transgenic model of a mouse where the axons light up in terms of fluorescence. So that gives us a truly 3D picture of the histology. And it's also possible for us to look at the vasculature as well as markers for astrocytes and microglia. And that really is exciting for us because it gives us a truly 3D representation of the structure that we can use to compare with our MRI. And that's really a powerful tool that we hope that we can use to validate our MRI microstructural biomarkers. Finally, just to touch on another opportunity, we're hoping to move to ferret MRI because the ferrets, they have much more white matter than rodents, and they mimic better what's happening with the gyroencephalic brains of humans. Really, we have an opportunity to do this at UBC because of rather mundane sort of reasons. And that's really the fact that ferrets, they're a predator species. And basically, if our mice start to smell any ferret smells, they get freaked out, right? So I can smell you, but he can't help it. And really what we had to do in the new facility is actually intentionally design sort of our rooms so that the sort of pathways for large animals and small animals were isolated. So we have a different prep room for the ferrets and other large animals and the small animals. So really the only place where they meet is the magnet, and hopefully the mice can't really complain if they're anesthetized. So yeah, that concludes my talk. So, you know, these are opportunities for us here at UBC, but certainly we're very much open to collaboration with other people. Yeah, just thank you for your attention. I'd be happy to answer any questions. Thanks.